Hello everyone. Welcome to our AMCP's e-learning day webinar. For today's the sections we're focusing on the advancing precision medicine in breast cancers, targeted therapies, and the role of managed care pharmacy. Thank you for taking your time for today to join us. My name is Jenny Lee. I'm a research assistant professor at the University of Houston College of Pharmacy, and I will be your moderator for today's webinar. Next slide, please. This continued pharmacy education sections was designed to achieve the learning objectives listed on this slide. This activity is supported by an independent medical education grant from AstraZeneca Pharmaceuticals, Daiichi Sankyo, and Merck Sharp and Dohme Corp. Next slide, please. The financial relationships reported by the faculty for these sections are shown on the slide. They are also available in your handout, which can be found under the Resources tab in Zoom. All AMCP education must be conducted in a manner that avoids the fact or appearance of conduct which may violate antitrust laws. Please refer to the antitrust policy located at the website listed here. It's now my pleasure to introduce our faculty for today's program. Dr. Ryan Hamtrick, the Vice President of Pharmacy at Emory Health and Winship Cancer Institute, and Dr. Laura Bobots, who is the Senior Vice President of Clinical Strategy and Growth at OncoHealth, and Dr. Marina Nikolaski, who is the Manager of Pharmacy Care Management at Health Plan Alliance and Henry Ford Health System. The full faculty biographies can be found within the handout. Now we will begin our presentations. Dr. Hamstrick, I will pass the floor now to you.
Now I get to kick off more of a formal presentation style where I'm really looking about sharing the power of genomics and really collaboration, meaning that we're taking the personalized breast cancer through molecular profiling, which some of the questions you've seen before, but also how do we integrate that through shared decision-making within tumor boards? Thank you, and let's transition to the next slide to get started. So we know that breast cancer is not all the same. When you see one patient with breast cancer, that doesn't mean the next person's going to have the same diagnosis or even treatment approach because of the heterogeneity that is needed. And we know within breast cancer, there's good innovation that's occurring because it includes that biological subtyping with different prognosis and responses to therapy. We now look at different treatment classification, such as estrogen receptor positive, progesterone receptor positive, HER2 expression. What is the tumor grading? Is it adjuvant, neoadjuvant, pre-surgical procedure? Is it metastatic? All of that brings in a unique treatment approach. And I think that we need to modernize the way that we think about breast cancer instead of just ER/PR positive or negative or HER2 expression, but now thinking about the genomic profiling, the molecular subtyping that plays such an important role when we're looking at breast cancer. And this means that precision medicine is helpful because it's a gatekeeper getting the right therapies and having the right oversight and prognostic treatment approach. So we're now taking that traditional classification that you see here on the left and modernizing it with molecular subtyping, genomic profiling, and now we might have 7 gene microassays. We might have 12 gene microassays. And this means that we can select the best treatment for that patient to get the best progression-free survival, overall survival, and now treatment sequencing. Next slide, please. So I really want to build upon what we just discussed. And I think it's really important to recognize that multi-gene assays, up to 70 gene micros, are going to be important because it can show you whether you might have high or low genomic reoccurrence. And also it might pick up on, whether you have early stage or late stage treatment. And this is really important, especially in those HR positive or hormone receptor positive patients, to make sure that we're doing the right selection up front. Because if we can improve progression-free survival, we want to do that now and make sure that we're doing it in subsequent lines of therapy. And that's where the next generation sequencing is being utilized to find what are those actionable mutations. We might have started someone on a CDK4/6 inhibitor, but when do we test again to see if there's another genomic receptor positive that we need to be worried about or actionable mutation? And here, an actionable mutation upon progression might be an ESR1 mutation. We might be really worried about PIK3CA for a patient to make sure that we're giving them the best outcomes possible. And then we know that there's pan-tumor expressions like NTRK and BRCA that we need to be concerned about. And as we think about that next generation sequencing, there's ctDNA that's giving real-time insights, not only into actionable mutations, but also resistance, and also allows us to check post-progression in a timely manner to make sure that we're customizing the treatment approach to create good stewardship for us as health plans, but also for the patient and the provider. Because when we have early treatment selection that's thoughtful and correct, it allows us to maintain a sequencing strategy that preserves the clinical efficacy and the safety. But on the end, we also can use clinical trial matching or even de-escalation of therapy as appropriate based on patient response. Next slide, please. So when we think about all this testing, some of us might think, well, if we have, comprehensive next generation sequencing, if we have RNA, we have DNA, how is all this really come into play? And I think it's important to understand that as we develop these molecular findings, as we understand somatic mutations like PIK3CA, as we look at germline mutations like BRCA1, BRCA2, this allows us to select specific therapies that otherwise may not be frontline to every practitioner. So if we are talking about that somatic mutation, this is when a PIK3CA mutation comes into play. We might use alpelisib plus fulvestrant for these patients, and we know that these patients will respond even better. It's now taking those genomic therapies that were once very concerning and bringing in a targeted therapy that we can leverage that unique genomic factor to drive efficacy, to drive target mutations on the tumor, and drive best outcomes. And in the same regard, we want to make sure that we're doing it at progression. So somatic mutation like an ESR1, we might pick up on that mutation upon progression. We might treat a patient with a CDK4/6 in the frontline setting. We will test, and if they're ESR1 positive, then can we move them to an oral SERD? And I think it's really important to understand that HR positive, HER2 negative patients are now having more and more therapies that are coming into play. And so we've got to be very thoughtful about how we're testing, when we're testing, but also the treatment approach that fits that unique genomic target. And subtype forms in chemotherapy versus endocrine therapy versus targeted versus immunotherapy. Even when we think about MSI high and DMMR and all these emerging, I would say, tumor mutational burdens that we continue to see. So at the end of the day, the biggest takeaway is personalized treatment improves outcomes, it minimizes overtreatment, and it allows us to provide the best efficacy for that individual patient. Next slide, please. So as we're developing this treatment plan and personalized approach, it does not happen just in a silo. More and more, we're seeing disease state working groups or tumor boards that emphasize the interdisciplinary and multidisciplinary value of interpreting genomic value. So now we have surgical oncologist, the medical oncologist, the subspecialist, the pathologist, the pharmacist, and even genetic counselors really formulating that treatment plan. We'll review genomic reports, make sure that we are having reflective testing based on how that patient's progressing, and also make sure that that genomic assays being utilized are occurring in the frontline setting. We can do this by working with pathology to validate the test. We want to align treatment decision-making with guidelines and patient goals. And that's important not just to the healthcare provider team, but the payer. Because if we're paying for genomic tests, we want to make sure there's value that can be pulled through, distilled, and lead to actionable treatment decisions. And also make sure that we're stewarding genomic targeted therapies to the most appropriate patient population. We also want to understand there's shared accountability and personalized care planning. And more after diagnosis and genomic testing, pharmacists are helping the care team identify what's the most appropriate treatment, not just in frontline, not just in second line, but in ongoing progression. And also, as we create more equity and more shared understanding of what testing to be ordered by what line of therapy, we create better health equity and more consistent care across the treatment continuum, across many of our covered lives and provider members. And I think this, at the end of the day, is one of the goals, is how do we bring it back to the patient? Next slide, please. So as we're thinking about the shared decision-making, I really like this visualization, that we have that patient that's sitting in the center. We have the payer, we have the provider, we have the pharmacist, the surgical oncologist, the medical oncologist, and we're looking to standardize and improve standardization across all treatment locations. It doesn't mean everyone's going to be 100% standardized in their treatment approach. But if we can work on creating consistent framework of what genomic assays are important frontline, what should be used reflexive in second line, and what treatments should be matched per genomic expression, we can implement best practices. Because now we're even looking at genomic and germline, but even looking at the expression of HER2, HER2 low, HER2 ultra low, triple negative, and therefore we can implement treatment decisions, medical policy that really reflects best in class care, but also pulls through this consistent treatment method so we can meet these medical policies based on the authorization for genomic testing, based on the treatment pathways that we have for oncology, and ultimately drive best in class care. So as we've talked about shared decision making, it's now time in the next slide to bring it all together. And so this is one of the most important considerations on the next slide is to talk through this. So if we transition now, we can now look at what the impact is. And I think this is what we're all working towards. It's alignment of formulary and treatment pathways, both genomic and medications, across an organization, across a payer network, so that we can leverage location and access to make sure that patient has coverage and has access to subspecialists. And even if they don't, if it's a community oncologist, if we're utilizing best-in-class care and medical policy, it can approach this genomic assay should be ordered in frontline. We can approve this targeted therapy as long as this test was ordered. And therefore, we create health equity and consistency of care among provider groups and even locations. We formalize tumor boards to make sure we're extracting the latest information from our surgical oncologists, medical oncologists, pathologists, genomic counselors, and pharmacists to make sure that we're bringing relevant information that's cost effective, but also best in class care. And we want to integrate that team into the decision making and even the payer arm and the pharmacy benefit, making sure that we have cross benefit communication. If we're dealing with a HER2 negative or HER2 positive patient, we might have a medical benefit, antibody drug conjugate, or targeted therapy, and we might have a pharmacy benefit product. How do we make sure that we're aligning policies and coordinating care? We want to use strong internal decision making and data to drive the sequencing of therapies and really work on this as we see more community oncology and hospitals continuing to align practices across the network. Because at the end of the day, a consolidated approach to treatment selection and formulary evaluation based on leveraging data, outcomes, and real world evidence will allow us to drive value, not only in a payer arm, not only in a health plan, but across an extensive network of providers. So thank you for hearing out that value proposition. And now I'm looking forward to handing off to my colleague, Dr. Laura Boboltz, as she continues to get even deeper into these novel biomarkers for breast cancer therapies.
Thank you so much, Ryan, for the fantastic review of the importance of genomic testing in breast cancer. So let me dive into some of the treatments we have that are biomarker targeted. Next slide, please. So if you look from the beginning of 2023, we have had 5 new therapies, FDA approved new indications that target biomarkers in breast cancer. First, starting with elacestrant for patients with estrogen receptor positive, HER2 negative, ESR1 mutated metastatic breast cancer, second line after endocrine therapy, followed by capivasertib plus fulvestrant, hormone receptor positive, HER2 negative metastatic breast cancer with either a PIK3CA mutation, AKT1, or PTEN alteration, also second line after endocrine therapy. Then we had one move to first line therapy, inavolisib plus palbociclib fulvestrant in PIK3CA mutated hormone receptor positive HER2 negative metastatic breast cancer patients. Next, datopotamab deruxtecan, hormone receptor positive HER2 negative metastatic breast cancer, about third line after your endocrine and chemotherapy, second line chemotherapy, third line overall, and trastuzumab deruxtecan, which we'll talk about coming up because this gained a new indication in hormone receptor positive, HER2 low or even ultra low metastatic breast cancer patients after endocrine therapy. But what's coming? That's the exciting part about what we do in the oncology world is we see new upcoming therapies all the time, right? So the pipeline is very vast. We're going to talk about camizestrant-based therapy, vepdegestrant, also imlunestrant plus or minus abemaciclib may be approved in the coming years. Imlunestrant single agent, perhaps, in ESR1 mutated previously treated metastatic breast cancer, maybe even in combination with abemaciclib, irrespective of biomarker. And then we'll also talk about trastuzumab deruxtecan, had data in combination with pertuzumab. And there was data from ASCO with sacituzumab govitecan and pembrolizumab, perhaps in the first line use of metastatic triple negative breast cancer. So on the next slide, what I want to do is dive into your biomarkers specifically, starting first with ESR1 mutations, because if you haven't heard about this, you're going to hear a lot more about it. So ESR1 mutations happen in up to 40% of hormone receptor positive HER2 negative metastatic breast cancers that essentially have been previously treated with an aromatase inhibitor and are developing perhaps resistance to that aromatase inhibitor. So it's an early indicator for us that the patient's cancer might be about to progress on that aromatase inhibitor if it develops an ESR1 mutation. Today, NCCN recommends testing for an ESR1 mutation by a liquid biopsy. So that's your next generation sequencing test done on the blood. And NCCN recommends to test for those ESR1 mutations via NGS in hormone receptor positive HER2 negative metastatic breast cancer patients after they progressed on endocrine therapy. So currently, we test for these usually second line or later. Today, we have elacestrant FDA approved in that second line or later space for patients with an ESR1 mutation, but there's therapies up and coming that we're going to talk about. So on the next slide, I want to dive in and take you on a bit of a journey of a clinical trial that was a lot of buzz at ASCO, our biggest oncology conference of the year. So this is an upcoming therapy that I'm anticipating to see approved in 2026, camizestrant, an oral SERD, in combination with a CDK4/6 inhibitor. And basis for that approval is likely coming from the SERENA-6 clinical trial, which studied patients that were receiving an aromatase inhibitor plus one of the three CDK4/6 inhibitors in estrogen receptor positive HER2 negative metastatic breast cancer. And they kept testing the patient's blood every 2 to 3 months. And when that patient developed an ESR1 mutation, then they were randomized to camizestrant, keeping that CDK4/6 inhibitor going, or just continue that AI plus a CDK4/6 inhibitor. But the key is that they did not have disease progression on the aromatase inhibitor. So you're trying to catch early molecular progression without disease progression on imaging. So on the next slide, just to show you a quick schema in the trial, started out over 3,000 patients, and you'll see in the purple all the way on the end, ended up with 315 patients. So roughly 10% of the population tested every 2 to 3 months in the blood for that ESR1 mutation, had an ESR1 mutation develop, and were randomized on the trial. It took a median of 21 months, though, until they saw an ESR1 mutation in those patients on an AI and CDK4/6 inhibitor. On the next slide, if we look at some of the results here, you can see that in the middle, there was a 16-month progression-free survival duration with camizestrant plus CDK4/6 inhibitor versus 9.2 months with the AI and CDK4/6 inhibitor. So that's a 6.8-month improvement in progression-free survival for a mid-first line switch, without disease progression, switching mid-first line when that ESR1 mutation develops. However, patients randomized to the camizestrant arm did experience a little bit more neutropenia, and they also experienced photopsia, which we don't hear of much in the drug space of a side effect. So photopsia is flashes of light in the peripheral vision. So we need to make sure our patients are aware of that. The great news, though, was adding camizestrant to the CDK4/6 inhibitor versus using the AI, keeping the AI going. It did improve the quality of life of the patients, and there was a longer time until deterioration quality of life with the camizestrant arm. So overall, on the next slide, just to sum up this trial, what we'll see here is that switching mid-first line to the camizestrant plus, keeping that CDK4/6 inhibitor upon ESR1 mutation, improved the progression-free survival for patients. We're anticipating a 2026 approval. We do have to keep in mind, though, that this does require repeat testing of the blood to look for that ESR1 mutation. These next generation sequencing tests looking at that ESR1 mutation perhaps in the blood can cost anywhere from $3,500 to $5,000. So we have to keep in mind the cost of repeat testing and the psychological burden on the patient. they're thinking, oh my gosh, what is in my blood now? Not alone, how are my imaging results going to come about? So very different type of approval if this comes about to switch the drug based on molecular progression. Also, keep in mind there is a Medicare NCD 90.2 that supports coverage of one NGS test in advanced solid tumors. But this strategy requires repeat NGS testing. So I'm really curious on the payer side if payers are going to adjust their policy, or how is support going to be had for patients to cover that repeat NGS testing. That will be very interesting to see how this plays out. And also to let you know that this did not improve survival just yet. That takes longer time. So the discussant of this trial at ASCO mentioned, is more time on this treatment worth going through the testing process if it does not help you live longer? So just something to ponder, because we do hope that this therapy in time we can see if it improves overall survival, but that data is immature. So moving on the next slide, switching gears to another investigational therapy, vepdegestrant. This therapy we're hoping to see approved in 2026. This therapy could be the very first oral PROTAC, which is a proteolysis targeting chimera estrogen receptor degrader. So different type of mechanism of action that we haven't seen before, but similar patient population. As we're looking for patients that have an ESR1 mutation, those patients are more likely to benefit from this oral PROTAC drug, similar to your oral SERDs. Now, vepdegestrant was studied in the VERITAC-2 clinical trial versus intramuscular fulvestrant, or IM SERD, in patients with estrogen receptor positive, HER2 negative metastatic breast cancer after 1 to 2 prior lines of endocrine therapy. Important to note, patients were required to have a previous CDK4/6 inhibitor, because those are part of standard of care first-line therapy for this patient population. But elacestrant, your oral SERD, FDA approved today for patients previously treated with that ESR1 mutation, that was excluded from prior use to be eligible for vepdegestrant. The agent did increase LFTs, so once this gets approved, we'll need to monitor liver function tests. We do need to look out for nausea and vomiting and diarrhea perhaps, but the good news is patients didn't require a lot of dose reduction. So this isn't a drug that's heavily dose reduced for toxicity or discontinued for toxicity. So not likely a drug that's going to be added to split fill programs on the payer side, if you ask my humble opinion. Efficacy: use of vepdegestrant in patients with an ESR1 mutation previously treated had a 5-month median progression-free survival versus 2.1 months for fulvestrant, for a 42% reduction in the risk of disease progression or death. So I'm expecting this agent to come to market hopefully next year with an FDA indication in estrogen receptor positive, HER2 negative, ESR1 mutated advanced metastatic breast cancer previously treated with endocrine therapy. On the payer side, coverage is more likely to align to label. Your ESR1 mutation will be required per the label, I believe. And also, some payers may consider requiring a CDK4/6 inhibitor previously in the coverage criteria, because that is standard of care prior therapy and was required in the trial. And some payers may have a limitation in their policy that if there's disease progression on elacestrant, we do not know how vepdegestrant will work from an efficacy standpoint. Okay, next slide, switching gears to HER2 targeted therapies. This is one drug, trastuzumab deruxtecan. We like our acronyms in oncology, we call it TDXD. And in the metastatic breast cancer space, this therapy has 4 different FDA indications today, and we're expecting another one to come sometime soon. So it's used in HER2 ultra low metastatic breast cancer that's progressed on at least 1 endocrine therapy in the metastatic setting. And in that HER2 ultra low, these patients have an immunohistochemistry of 0 on staining, but they have some membrane staining. So they have Maybe just one cell has a little bit of HER2 there that's striking positive. So they're having some staining. It's not goose egg, negative, no staining. So from a managed care standpoint, keep an eye on the pathology because people report this differently. It's also approved in that similar setting in HER2 low disease, which is immunohistochemistry of 1+, so a little bit more staining, or immunohistochemistry 2+, but uh, not positive on the ISH fish, I like to call it. It's easier to say. Your fluorescence in situ hybridization or your ISH in situ hybridization. If your fish or ISH is positive, you're HER2 positive, and you get therapy per the purple FDA indication to the right. If you are ISH negative and immunohistochemistry 2+, we call you HER2 low. And in that case, the therapy is used either if you're hormone receptor positive as second line after endocrine therapy, or if you are any hormone receptor status after also the patient has received prior chemotherapy in the metastatic setting or recurrence within 6 months of curative endocrine therapy. But the cool stuff is what's up and coming on the very bottom. So hopefully we'll see an FDA indication for trastuzumab deruxtecan in combination with pertuzumab as the double therapy in first-line HER2-positive metastatic breast cancer. So on the next slide, I'll highlight this trial that we saw at ASCO, the Destiny Breast 09 trial, which found that the combination of trastuzumab deruxtecan plus pertuzumab significantly improved progression-free survival by an absolute 13.8 months. 13.8 months, large benefit over our previous first-line standard of care in your classic HER2 positive patients. So that's the FISH positive, the ISH positive, the patients that have an IC of 3+. In this patient population, our previous standard of care, and still actually is today, is THP, or the Cleopatra regimen. Taxane, docetaxel, or paclitaxel with trastuzumab plus pertuzumab. So here, your trastuzumab deruxtecan plus pertuzumab improved progression-free survival over that taxane, trastuzumab, and pertuzumab-based regimen. But we do have to keep in mind that trastuzumab deruxtecan, it's a monoclonal antibody with chemotherapy attached to it. So these patients had a median duration of 20 months of trastuzumab deruxtecan in combination with pertuzumab, and that's 20 months of constant chemotherapy. Where on the current regimen today, first line of taxane, trastuzumab, pertuzumab, we usually drop the taxane after 6 to 8 cycles, and the patient continues on dual HER2 targeted therapy alone, plus or minus endocrine therapy if they are hormone receptor positive, but no chemotherapy in our current regimen of THP for that maintenance portion to maintain your response. So keep in mind, I am curious how this trastuzumab deruxtecan plus pertuzumab will affect quality of life. This therapy did receive a breakthrough therapy designation. It does not have a PDUFA date yet, and currently it is off-label. We do not have coverage support in NCCN just yet as of August 13th. We don't have coverage also per a full text publication just yet as of August 13th. So for my managed care friends, we're really waiting for that. And then keep in mind on the managed care side that if this therapy does get approved, trastuzumab deruxtecan plus pertuzumab, if the patient is hormone receptor positive, the trial did allow the addition of endocrine therapy. So you're talking endocrine therapy like an aromatase inhibitor, not endocrine therapy plus CDK4/6 inhibitor. That could be overtly toxic. We do not know that combination today's efficacy. On the next slide, let's switch gears to our last mutation I have for you, PIK3CA mutation. Another one that happens in up to 40% of patients, so easier to remember, similar to ESR1 mutated patients. In those that are hormone receptor positive, HER2 negative. Now here, NCCN recommends testing for PIK3CA mutations via the next generation sequencing or PCR tests in patients upon metastatic or recurrent disease that's hormone receptor positive, HER2 negative. Today we have 3 regimens there that target PIK3CA mutations, and the one thing I wanted to highlight is we had new news recently this year that the 3-drug combination of inavolisib, palbociclib fulvestrant actually improved overall survival by an absolute 7 months in that first-line setting of patients with a PIK3CA mutation that were hormone receptor positive, HER2 negative, and have received previous adjuvant endocrine therapy. So on the next slide, just summarize, it's like, I'm going to show you some stuff. Get your glasses out or get real close to the camera. There's a lot here. Don't worry, I won't talk your ear off that much, I hope. So here are 3 regimens FDA approved today in patients with a PIK3CA mutation. I mentioned the inavolisib, palbociclib, and fulvestrant, very key with that overall survival benefit. These drugs a lot of times can cause hyperglycemia, so that needs to be monitored. The next one in the middle, capivasertib in combination with fulvestrant, is used in that second line space with a PIK3CA mutation, or there's always an or a but in oncology, right? We love our nuances. Or if the patient has an AKT1 or PTEN alteration. So it can be PIK3CA mutation, AKT alteration, or PTEN alteration for eligibility. Overall survival data is immature, though, with the capivasertib-based regimen, and this can cause some diarrhea. And the last one, like Ryan mentioned earlier, alpelisib in combination with fulvestrant, this is another second-line or later regimen in patients with a PIK3CA mutation in hormone receptor-positive, HER2-negative metastatic breast cancer. We love all those crazy biomarkers. But unfortunately, in the SOLAR-1 clinical trial, alpelisib plus fulvestrant did not improve overall survival. So that's key because we're trying to think from a managed care standpoint, what's the value of these therapies for our patients? Okay, and with that, next I am going to pass the torch to my amazing colleague Marina to talk about managed care considerations with genomic testing.
Thank you, Dr. Bobles. Thank you for your time. Thanks for joining us here. I'll finish recapping some of the concepts that we heard today and talk about some of the things we can do from a managed care pharmacy perspective to help bring these precision guided therapies to our patients and members. So first slide, please. Okay, so I wanted to talk about considering the audience is the rationale for the drug coverage that we consider when we talk about medication coverage. Of course, with the FDA guidance, we have the regulatory approval, which show us efficacy of medications. We also consider cost effectiveness and patient preferences. So how do we contrast this to biomarker coverage? Next slide, please. So we have really high correlation between what happens with drug coverage and biomarkers. So drugs, of course, we have to make sure that these are pharmacologically active entities. And with biomarkers, we consider the validity of the test. Where with drugs, we talk about effectiveness so that they have an actionable target. With biomarkers, we have to consider clinical utility. And where with drugs, we consider safety. With biomarkers, we have to considering that they bring information to our members. And I think one of the earlier presenters talked about potential psychological harm that can be brought with some of the knowledge without an active target. We have to consider harm avoidance. So next slide, please. So unfortunately, with biomarkers, unlike with medications, we do not have the assistance of our regulators. So the FDA has taken an enforcement discretion approach to regulating biomarkers. Although they do have the statutory authority to do that, the only segment of the biomarker market that is FDA regulated are the test kits that reach our patients. However, when we're talking about samples that go to be tested in the lab, they're not FDA regulated. They may be submitting some of, as you can see shortly, some biomarker assays can get pre-approval from the FDA. However, labs are typically regulated by CMS, and they're regulated on the analytical validity basis only, just to make sure that they produce true results for the test that they're producing. FTC is the third part of the alphabet soup, and they, of course, regulate direct-to-consumer messaging that, just like medications, biomarker assays also produce. So with all the three entities, unfortunately, we still have gaps from the regulatory standpoint. But of course, as you may have heard, there are a lot of changes at the FDA. That approach may be changing going forward. However, at this time, we are, as you can see on this slide, NCCN is one of the guidelines that recommend specific biomarker or genomic assays. And and I'll just group the Oncotype DX together, out of the 5 different products they recommend, only 2, Mammaprint and Prosigna, have received FDA pre-approval. Thank you. Next slide, please. Thank you. So without having a regulatory body to vet the evidence for genomic testing for us, we do, of course, have guideline to guide us. Here, talking about the 3 guideline entities that talk about tying genomic assays with therapeutics. Of course, we have the NCCN. This is a very high inclusivity, frequently updated guideline, and many of us, of course, are familiar with that. ASCO, perhaps a more conservative body when they produce their recommendations, they balance that inclusivity with the clinical utility. And the most conservative out of the 3 is the ESMO, the European Society of Medical Oncology. They, of course, produce recommendations based on the SCAT scale, which they have validated as a tool for their recommendations. So they were weighing really heavily the targets that produced increased survival. However, as we can see on the next slide, when we talk about invasive breast cancer, the guidelines have a really high level of concordance. So in breast cancer, again, as previously stated, there's so much evidence to do precision-guided therapeutics that essentially all guidelines are listing every genomic assay that is available, with exception AGCC, of course, is the staging body. The only genomic assay they have validated for staging is the Oncotype DX. St. Gallen's does not mention specific products. The rest of the guidelines, as you can see, have really high concordance in their recommendations. Next slide, please. Thank you. And of course, when we talk about, as we can see in precision medicine, as soon as you put something on paper, it may become outdated. But here are some examples of biomarkers that are just individual targets that have companion therapeutics available on the market. And of course, there are recommendations across guidelines for these individual biomarkers as well. Next slide, please. We also have high level of concordance when we talk about repeat testing. And of course, that's not perhaps speaking about the recently mentioned method of retesting members to see if mutation had appeared. This is talking specifically about a specimen at a same point in time using multiple assays to understand, to guide therapy, essentially. So in Optima Prelim trial, while all assays were cost effective, unfortunately, there was lower than 40% of concurrence in the classification by multiple tests. That, of course, can cause a lot of confusion to the member and the provider. Guidelines do not recommend. So a guideline recommended product should be used, and that should be what will be guiding a future therapy. Next slide. Okay, so just restating the coverage principles that we can use for biomarker test, of course, understanding that there should be established clinical and analytical validity, and as well as clinical utility. So we have to consider how tests will guide therapy, potential for harm that they can create, and whether, as we see with Oncotype DX, avoiding cytotoxic chemotherapy in certain situations. So in a sense, this is very similar to how we consider medication coverage as well. And now I will shift gears on the next slide, and I will talk about some coverage, broad coverage principles, as well as some additional things we may see in our interaction as managed care pharmacists. So again, restating, we have to bridge the gap. We have to consider which biomarkers should be covered. We have to work to improve access, and this is essentially a multidisciplinary effort. And I'll talk about it in the upcoming slides. What can we do? Who do we work with to make sure that the precision-guided drug gets to the patient in as short time as possible? And of course, we have to consider what value, what barriers, disparities, and different patient factors can be causing our members, and work to overcome those as well. Next slide, please. So access to coverage, of course, as we talked about, there's a high level of evidence to provide genomic testing, but there's no federal mandate to provide coverage. CMS, of course, provides a genomic assay coverage. Commercial plans do have discretion whether to provide that as well. In certain states, legislation has been introduced and sometimes passed that states that genomic testing has to be covered. There is some variation. It can depend on whether the health plan is state governed, such as Medicaid, or state employee health plans, such as in Florida. And some states mandate that specific disease state genomic testing has to be covered. Of course, oncology being first and foremost out of all disease states where precision medicine is used. Next slide, please. So unfortunately, although coverage is widely available, a recent study by the American Cancer Action Network, and this was specifically querying oncologists with regards to BRCA genomic testing. And an unfortunate 66% of oncologists reported that there are barriers to their patients in getting appropriate testing. In part, this may have to do with how reimbursement is structured for clinical, based on the clinical fee schedule, and how CMS crosswalks and sometimes fills in the gap. However, when we're talking about this quickly developing area of next generation testing and other testing methodologies, the reimbursement just has not caught up. So we, as health plans, do have to be aware of that. And I'll talk in the subsequent slides how we need to collaborate with others to overcome these gaps. Next slide, please. Okay, and Dr. Hamschild mentioned molecular tumor boards. I wanted to bring this here to talk about why, although we're very much guided by FDA label, by guidelines, why we may sometimes see more progressive approaches than we see in other disease state with in oncology and breast cancer specifically. So of course, use of molecular tumor boards is employed when patients are resistant to previous lines of therapy. It is really hard to do large-scale studies. So this study by Miller et al. Showed kind of a proof of concept for molecular tumor board-assisted care versus gauging how that improve potentially progression-free survival over previous lines of therapy. So the primary outcome was to see whether progression-free survival was increased by 30% in second or greater lines of therapies. Next slide, please. And patients were seen with a wide range of tumors, essentially representing incidents that we see in the community. And remarkably, almost 60% of patients achieved 30% increase in progression-free survival with subsequent lines of therapy. And of course, typically we see that a lot shorter when subsequent lines of therapies are employed. It's usually a reduction. It's a single arm study, so while we see some secondary parameters, they were wide ranging. There were a few deaths, but this really provides a proof of concept why in managed care we may see some therapies that are recommended by these bodies. We need to know how to engage them, how to get readout from them, and approach this in a collaborative manner. Next slide, please. Another entity that we're likely to interact with are precision medicine stewards. So in some community-based oncology programs without academic center resources, we see these essentially point people getting engaged. They are they can QA the specimen. They have shown to decrease the time from sample to test by almost half. They, of course, can assist with prior authorizations, which can be a point of interaction for health plans, and that's somebody we should be aware of. Okay, next slide, please. Thank you. So as you can see, there are multiple facets in precision medicine. We, in managed care pharmacy, and in my health plan in particular, we interact with clinical geneticists and medical directors all the time to understand the readout for molecular tumor board, to understand why a specific genomic testing was used. One feature that we're considering and should be considered is the preferred testing site, where out-of-pocket costs can be overcome for our members, as I mentioned, that being a barrier very frequently. And it can also increase the time, decrease the time to see fast results and take next steps to bring the drug to the patient. Next slide, please. Okay, and tying it all together, of course, collaboration between health plan and a specialty pharmacy that hopefully has access to the preferred testing site. Our goal is to, of course, bring the drug as quickly as possible to our patients. Again, multidisciplinary approach, interacting with other clinicians in the plan, potentially contracting department and specialty pharmacy. And all of this is, of course, with the patient in mind. And at this point, I'll conclude the presentation and take your questions.
There's one question from the audience. It's about the Medicaid cut. So the question is, how well the pending Medicaid cuts gonna affect the efforts for equitable access to the therapies? I'm gonna pass to our presenters to address this question.
I'll jump in first, if that works okay. And, thank you for the thoughtful question, because I think at the end of the day, what I was seeing is we want to make sure that health equity is maintained. And so as we have more cuts, how is that going to impact our patients? So thank you, TJ Cook, for your thoughtful question. And yeah, I think it's a real consideration. I think we're still going to be able to uphold medication access to innovative therapies. I know that Medicaid and the administrators are always working hard to make sure that we have best evidence-based. But I think as cuts still come, what that does is it leaves some gaps in patient insurance and the affordability. And I think that's really where we come around them to say, how do we get them enrolled? What are alternative options? And as we're working on the Medicaid formulary, how do we work on cost effectiveness with the adoption of innovative therapies, but also upholding some of the tried and true therapies that have great efficacy in current state? But I think it's a key consideration as we move forward to make sure that the formularies can grow with the innovation, but at the same time, we can also get more and more patients enrolled so they are covered and they get access to these, in my opinion, life-changing therapies for breast cancer.
Okay, great. And I also have several questions for our faculty speakers. So my first question is regarding the payer side. So I want to ask all of you, are you seeing or have you ever observed any confusion on the payer side with the application of precision medicines in breast cancer?
Sure, happy to start with that one. Thank you, Jenny. One, I see some confusion with reading the next generation sequencing report. Perhaps reading it on the provider side, is this really a mutation that we have a targetable drug? And then just us reading it on the managed care side, because sometimes we just get a scanned in paper that's got dark all over it and we can hardly see it ourselves. So I think that's one of the challenges, is matching the unique mutation, alteration, aberration, next generation sequencing report to the appropriate drug. And sometimes we see on the managed care side, people want to push the mold a little bit because perhaps the report says there's a drug that targets this mutation, but it might be a drug that's approved in a different cancer that I can't find a publication to support that use in the cancer they're requesting it in. It's not FDA approved in the cancer, and it's not in NCCN's compendium for the cancer being requested. So just to tie back to the molecular tumor boards, Marina did a fantastic job talking about those. Ryan as well at the beginning. Sometimes they make me pull my hair out though, because I'm like, yeah, it's fantastic a board of people looked at this, but you're recommending a drug for because of a biomarker, but I don't have coverage support from a managed care standpoint. I don't have an FDA approval. I don't have its support in NCCN. I don't have full text publication. So I think if we can just talk with our clinicians on the provider side, bridge that gap, educate them about what suffices for coverage, that might help.
Great, thank you for your answer. I have another question from the audience. So patients and breast cancer nonprofits want to get involved and offer their input or patients-wise to the policy development. So how might that happen to support the managed care pharmacist?
Thank you for that question. Actually, there is a method to do that, and CMS, starting in 2026, is mandating that pharmacy and therapeutics committees have patient representatives on their roster, and we're very excited in my health plan to welcome some of these. They, of course, there are some qualifications. They have to have some understanding of therapeutics, however, definitely be representing the patient perspective. And I think that that's a really valuable methodology to get engaged. Of course, that's a very, as we're learning and with molecular tumor boards and features that come really almost daily in, from the evidence side, there's also the decision making at the level of the plan that happens after. We also engage in our coverage decisions with oncologists all the time. We have essentially expert panel that we deal with from the oncology pharmacy and therapeutics committee perspective that we reach out to almost daily. We speak to our on-staff geneticists a lot more than we used to as well. So I think starting with the P&T and then potentially getting engaged in the provider side and having that advisory role is really valuable.
Great, so thank you for the great addressable answers. And then I think considering the times, I would like to thank everyone for taking their times to joining us, and then also want to thank our awesome, great faculty speakers. And I want to say this concludes today's webinar, and we want to thank you again for being here, and we look forward to seeing you at the next sections. Have a great rest of the day. Thank you.
Thank you.